thank you very much for your time. Capricor is a clinical stage biotechnology company with 3 products in our pipeline. Today I'll be focusing on our 2 products in the regenerative medicine space, both platforms, and I think you'll agree by the end of it that we have a really exciting opportunity coming up. So we are a publicly traded company. I most assuredly will make forward-looking statements. Please forgive me for that in advance. So the foundation of our cell therapy program was our CADUCEUS clinical trial. It's a proof of concept in humans trial in patients that had a large heart attack. And then we were given them their cells down their coronary artery and waited 6 months and then a year where we did magnetic resonance imaging and studied the effect of the cells on their hearts. And what we found are 2 things. First, in this groundbreaking study where we were the first to put a coronary-based cell down a coronary artery, we found that it was safe to do so. Not a problem there. But we also found that there was a significant reduction in the amount of scar that was in the heart at the end of the study. So the patients had less scar, which we think would be clinically relevant if we were to follow them long term. And we also had an increase in viable mass. That was the healthy beating tissue that was in their hearts, replacing the scar that had been there. So this led us to move forward with a very active clinical program with these cells. They're called the cardiosphere-derived cells. CDCs, or now their product name is CAP-1002. Based on the CADUCEUS data and a tremendous amount of synergy, J&J, through their partner Janssen, made an investment in Capricor, both at the financial level but also in the clinical and manufacturing development level. So they paid us $12.5 million up front for an exclusive license option. That means they're going to track our progress through our all-star phase 2 clinical trial, which is ongoing right now. And at the end of that trial, they'll be able to decide whether they want to bring that therapy in-house. If they bring it in-house, and I think we all agree that doing trials in heart failure or heart disease are incredibly expensive due to the size of the trials that have to be done on a global basis, it's nice for a small biotechnology company to have a big pharma partner to carry that forward. And so to that end, J&J would then step up to the plate and take over all those development costs, pay us milestone-based payments in turn up to $325 million, which don't include sales, milestones, or royalties, and most importantly, take over also the commercial stage manufacturing. However, not to be a company that allows grass to grow under their feet, either J&J nor Capricor, we're working together actively at this time, building together a full CMC package such that we can take this therapy to pre-commercial stage should they bring it in. So this is an active collaboration that's ongoing as well as a long-term opportunity for the company. Shown here is our pipeline of the opportunities that are available in Capricor today. As I mentioned, our All-Star is our phase 2 clinical trial. I call it CADUCEUS with a kick. For those of you that have heard me present before or have followed the company, you remember that CADUCEUS used an autologous-based cell. That means we took tissue from your heart, we took it back to the lab, grew it up into a dose of 25 million cells, and infused it down your coronary artery. We found that was an effective strategy in terms of the therapeutic outcome, as you saw from the CADUCEUS data, but we felt from a business model and strategic development point of view, it was just not an effective strategy. So we decided to move forward with an allogeneic platform. So All-Star is using our allogeneic cell. We've reported out phase 1 data, which showed not only was it safe, but it was trending in the direction of the CADUCEUS data with a reduction in scar size, increase in viable mass, and trending towards improved ejection fraction. I use the word trending not because we didn't see significance, but because it was an open label trial without controls just to demonstrate safety of that allogeneic product. And so we can't really say that it was significant, but we sure think it looks pretty promising. And we're looking forward to the All-Star Phase 2 results in late 16, early 17. Dynamic is our trial, which we should be reporting out at the American Heart Association in advanced heart failure. Since we know that heart failure, now your heart is globally damaged, so instead of just infusing down one coronary artery, we're using what we call triple vessel infusion. So we're infusing down all 3 coronary arteries. We did a dose escalation study. That was also an open label study, 14 patients. That data, again, will be presented at American Heart, and we're looking forward to those results because it provided the foundation for our HOPE trial. I'm going to spend a little more time on the HOPE trial this morning because I think it shows 2 things. One, it shows that Capricor is now beginning to look at treating orphan diseases with cardiac implications, and also because it's so important from an unmet medical need perspective. So those of you in this room probably a few years ago only remember Duchenne muscular dystrophy from the telethon that was on Sunday mornings, and if you're old like me, that was all that was on TV, so we didn't really like it very much. But thanks to the parent lobby, that has been very active. We now know that Duchenne muscular dystrophy is this tragic, almost always fatal disease in young men that is characterized by a skeletal muscle myopathy that leads them ultimately unable to walk, to breathe effectively on their own, and as I said, leads to their death. What has been largely ignored, and I think you'll see in those quotes that I show at the bottom, is the lack of attention that was paid to the cardiomyopathy. The heart is a muscle as well. And so what we really began to understand is that as the skeletal muscle was dying, so was the cardiac muscle. And ultimately, most of these boys die from complications of the cardiomyopathy, especially in these days where the therapeutics for the respiratory complications are so much better. And so we saw a unique opportunity because I just want to draw the connection here between the cells that we have, which we've now shown to reduce scar in the heart, and then we have this paradigm where we have boys with increasing scar in their heart. It seems like a perfect match. So we went forward and did some preclinical work using what would be considered the standard model called the Duchenne mouse, or the MDX mouse. And what we showed here is that in the littermate control, shown in the yellow and the red bars, if you treat with cells, you see a significant improvement in ejection fraction. That's the ability of the heart to meet the needs of the body. The blue line were the standard mice, lab mice, regular guys that are just running around in their cage and have normal ejection fractions. Now, as you can see, after about 3 months, the effect begins to drop off. And so we wanted to make sure that we could continue to provide support for these failing hearts. And so we went back and did another study with a repeat dose. And what we show here is that if you repeat dose, you can reinvigorate the effect and actually, again, drive for the improvement in cardiac function. So 3 months in a mouse, which has a lifespan of about 2 years, is X in a human. We'll find that out as we proceed into the clinic, which we are now going to be doing very, very shortly. The other piece of data that I think is of real interest here is the improvement in exercise capacity in these mice. So again, same groups of mice, red and yellow are littermate control, MDX mice, blue are happy normal lab mice. And what you see is that the mice who got the cells can run longer week after week on a treadmill compared to their littermate controls. Now, is this just due to improvement in their cardiac function? It's possible. There also could be synergistic improvement in cardiac and skeletal muscle. We are now investigating that on the academic side. But suffice it to say, if you can run longer, you absolutely feel better, and that's what's important. So taken together, this data, along with significant amounts of other data showing the mechanistic implications of these observations, which is the invigoration of the mitochondria, basically the power generator of the cell, we are now about to begin, and it should be in the next couple of weeks, the HOPE Duchenne trial, 24 patients, one-to-one randomization, triple vessel infusion, and we're going to be looking for safety, of course, and hints of efficacy both at 6 and 12 months in these boys with cardiomyopathy-associated Duchenne muscular dystrophy. So you hear a lot about cell therapy and gene therapy, but what I'd like to talk about is what we think is the next generation of regenerative medicine platform, which is the exosome. Exosomes are made by cells. They're made by every cell in the body. They are the way that cells communicate with each other. They're nanometer-sized lipid bilayer vesicles packed with messenger RNA, microRNAs, yRNAs, and a variety of other peptides and proteins that go from one cell to another and tell them not only what to do, but also change actually gene expression by changing what happens in the nucleus of those cells. Because of their large opportunity, there's broad therapeutic availability. So you can use these in almost any disease in which you think inflammation or fibrosis might be an issue. So, we are moving forward with our platform therapeutic in the exosome space. What I can say is after working with cells for many, many years, they're much easier to work with. We can lyophilize them. We can store them on a shelf. They can be pulled out and resuspended and used in any variety of diseases. We're using the exosome because of the large opportunity that they provide outside of any specific location in the body. We're going to be advancing outside of the cardiac space. And we haven't announced the indication with which we're going to be moving forward with our exosome program at this time. But what I can tell you is that we've recently brought in an ophthalmologist to run our new therapies division. And so it's likely that we're going to be developing in the ophthalmologic space, which we're very excited about taking this step right outside the cardiac arena. And stay tuned for the announcement of that relevant milestone in the early part of 2016. So shown here are our milestones. We had a busy first half of 2015 where we completed all of the goals that we set for ourselves. Sundaritide is our natriuretic peptide program for advanced heart failure. Those are folks that are coming out of the hospital and are high risk for going back in the hospital. It's not specifically in the regen space, so I didn't talk about it today, but we're excited about moving forward with that clinical development program. We got orphan designation for our DMD program, and we submitted our IND for Duchenne muscular dystrophy, got that approved, and as you now know, we're starting that trial soon. We expect to have a busy fourth quarter with the initiation of the trial, reporting out of the dynamic results. As I mentioned, that's our advanced heart failure trial, and then, of course, announcing the first indication for exosomes and powering that very exciting therapeutic forward is going to be keeping us busy in the first part of 2016. So shown here on this slide are the people that helped take these therapies forward. Some of my team are here in the audience today, and what I can say is that it takes a really tremendous amount of brain power to get baby biotechnology off the ground, and I think we are in fact doing that. So I think that the people that are here to support me only show how smart I am at recruiting the very best that there is. I also want to take the last minute to thank the Alliance. This meeting has gone from a very small boutique meeting with a small group of attendees to really a large audience of diverse types of people from all walks of the field. And so it's exciting to be able to share the progress that we're all making with such a good group. So thank you for your time, and I look forward to talking to you out there if you have any further questions.